it's my absolute pleasure to introduce our second plenary speaker of the conference, Dr. Viviana Gradnaru from Caltech. Viviana grew up in Romania, and she came to the States to finish her bachelor degree studies from Caltech. She received her PhD from Stanford University. If I were to think of a modern model of a modern neuroengineer, it would be Viviana. She's made some incredible contributions toward many technologies that I think many of you use, optogenetics, tissue clearing, and more recently, she's really been a pioneer in driving the development of vectors for precision brain cell access, one of the big focuses of the BRAIN Initiative moving forward. And she's applying these tools toward understanding brain circuits, brain-body connections, as well as helping us understand better models for disease. And you're going to hear a lot about that. She's the recipient, not too surprisingly, of many awards. I'll just list a few here. She's received the Presidential Early Career Award, the NIH Director's Award, as well as the Vilcek Prize, just to name a few. So without further ado, Viviana, please. Thank you.
Good morning, everyone. And it's so special to be talking about the tools that my group and collaborators has been developing over the past decade, because as I was starting my brand new lab at Caltech as an assistant professor, the BRAIN Initiative was getting off the ground. And I remember being here at some of the first few meetings with a poster and trying to recruit students and trying to point them towards what I thought were barriers of all sorts in maximizing the benefit of neuroscience knowledge and technology for human brain disease. And that's why today I want to focus on something that's been building up over the past decade in terms of understanding the body's defense mechanism, and in particular transport across the blood-brain barrier, in order to move towards targeted and non-invasive study and repair of the brain. And as John mentioned, what focused me on this problem at first was my prior work in neurotechnology, such as optogenetics or tissue clearing. And I have to say, now in neuroscience, we have this bounty of research tools. And the experiments that can be done in a transgenic animal, in a rodent, are very complex and inspiring and very informational. And that's because of all these tools that you in the room have built over decades of very hard work, and internationally as well. And this is in very stark contrast to what we can do in the non-transgenic brain, in non-traditional research animal systems, and we've seen the movie from the salamander, right? We can learn a lot about regeneration that could inform how to approach neurodegeneration from such species that can have unique abilities that mammals do not have. So the big problem is, how do we take all of this bounty of tools that we have for transgenic mice and deploy them in different model organisms, including non-human primates and maybe towards human? And this is where it became apparent to me that because both the natural and disease behaviors, they're not localized in one particular cell type or brain area, aside from very few examples, right? Most of them emerge from activity in distributed and molecularly diverse circuits throughout the brain and the spinal cord. And when these circuits malfunction in any spot of the brain, you get the disorders that affect more than 1 billion people worldwide. And this is a severe underestimate, because I didn't include substance abuse. If we think about addiction and substance abuse post-COVID, it gets to a toll of 2 billion people. And in order to understand and restore brain function, we actually need brain-wide access to deliver these genetic tools and non-genetic tools that the community has been developing, and to act on the learning from efforts such as the BRAIN Initiative. However, there is a big challenge, because the adult brain is very hard to get to without surgery. You need to drill the skull, insert a probe, inject the material. And at the same time, there's the contrast that each one of our brain cells is within 20 micrometer of a vascular supply. You have this natural route that can reach each and every cell, neurons, glial cells, endothelial cells in the brain. And in total is about 400 miles of vascular highway, or 650 kilometers, depending where you're traveling from. And this would be a great opportunity to use this vast highway to deliver research tools and therapies. However, the blood-brain barrier is a physical, cellular barrier that blocks access. It's highly selective. So you have endothelial cells forming tight junction, reinforced by astrocytic enphys. And the blood-brain barrier allows nutrients and immune defenses, but at the same time is very good at blocking pathogens. That's why most viruses and bacteria cannot gain entry. And that is a really good thing. But that also means that we could not deliver all of the tools and therapies that we want. However, there is some hope because specialized blood-brain barrier receptors can enable selective permeability of blood circulating agents. And one example, like just an amazing success story, is coming from the use of viral vectors to deliver genetic material across the blood-brain barrier throughout the brain and spinal cord. And this was at the root of the therapy for children suffering with spinal muscular atrophy. Children like Emily can now run around, play, do things children are supposed to do rather than having their muscles wasted away. What was used for Emily and children with spinal muscular atrophy is a viral vector known as the adeno-associated viral vector serotype 9. There is a catch. AV9, for short, crosses enough and transduces enough neurons in young mammals like Emily, but it has a very hard time doing this in adult mammals. And once it crosses, transduces mainly astrocytes. So this focused me on the limited knowledge we have around blood-brain barrier transporters and how can we, one, increase that knowledge, and second, make tools that can allow delivery of genetic and non-genetic tools. And as a bit of background, there's only a handful of known receptors, and maybe the best known one is the transferrin receptor. As an entity, as a molecule was introduced around 4 decades ago, and it's been explored for blood-brain barrier crossing for about a decade with work from Genentech and then the Denali team. Transferrin receptor is present throughout the body and has a very important physiological role in iron transport. We could benefit from having many more solutions with diverse properties in order to expand the reach of crossing the blood-brain barrier. However, when I started this work about a decade ago at Caltech with my group and collaborators, the knowledge about receptors was not there. We had transferrin and a few others, but that didn't allow us to find a way to cross the blood-brain barrier across species very efficiently. So what we did at first was to take mechanism agnostic approach using directed evolution, and I will go through it one by one. And over years, we developed vectors that can cross the BBB from rodents to non-human primates. And once we've had these vectors and disseminated them, we've been using them both to apply them to disease models for brain disorders, but also importantly to identify how they work, the blood-brain barrier transcytosis pathways and cell entry mechanism, and this is in progress. And then, of course, this informs to target these receptors for rational engineering of such delivery vectors that can have more predictability and cell type specificity, but also going beyond viral vector delivery. And these pathways can then be recruited to understand and may also, with a lot of work from the entire community and investment, prevent and cure brain disease, including, importantly, for infectious agents, because once you know the weak points of the blood-brain barrier, you could also block them to infectious agents. And it's important to know that the path that I will describe next can be applied beyond the brain. It can be applied to understand other physiological barriers, placental, blood-brain barrier, skin, etc. Without knowledge of diverse receptors to cross the blood-brain barrier, the first effort was to use directed evolution to start from the AAV9, the one that's approved and used in the clinic for spinal muscular atrophy, and put the AAV9 through a directed evolution of proteins pipeline, which entails generating a lot of diversity. You take AAV9, and through cloning, you can generate libraries that theoretic diversity can be 1 billion, but with biological loss, you end up with tens of millions. And then you apply these libraries to the bloodstream of living rodents. And you apply selective pressure. And this is a very, very important step, because to raise above the biological noise in such libraries, you need a very clever stringent system. And the stringency of the system was figured out by Dr. Ben Deverman, now leading his group at the Broad. Once this is done, you can extract the brain and see who made it through. Which one out of these millions and millions of modified capsids can actually make it to the brain? And we were very happy to see that you can indeed go from AV9, that's approved in the clinic for SMA. However, you can see that the adult mouse brain is not transduced very well, to PHP-B first, and then with further refinement for production and effectivity, you can get PHP-B that delivers the product to the entire brain. And of course, in these libraries, you can also get vectors that can transduce the peripheral nervous system. What's important is that what's modified is the capsid shell of the vector, so the experimenter does have full control over what's packaged in the capsid. And that then can intersect with all of the other wonderful discoveries from the Brain Initiative in terms of enhancers, driver lines, microRNA target site knowledge, so gene regulatory elements, to target areas such as surgically very difficult dorsal raphe, under the aqueduct. This is a difficult surgery if you puncture the aqueduct. So you can access it non-invasively through a simple bloodstream infection. This is important. Dorsal raphe is implicated in many neuropsychiatric disorders. You can target the midbrain, deep nuclei, dopaminergic nuclei involved in Parkinson's, or cerebellum for ataxia. All of these are very difficult surgeries. Another interesting application can be that you can capture very large brain areas, for example, to screen for gene regulatory elements instead of generating transgenics or doing spot injections. So this is an example of using the DLX enhancer from Gord Fischel and also Vikas Sohal's work, where with the DLX enhancer, you can capture the inhibitory forebrain neurons, and the inhibitory neurons have a big role in epilepsy. So over the years, we've been developing these tools and apply them within the lab at Caltech and also with collaborators in the US and internationally, to problems such as gut motility and microbiome, to pain by modulating DRG activities, or for neuroscience. And those experiments taught us a few good things about our tools and a few still to improve and to understand items, which I will focus on today. When you inject the capsids through the bloodstream, they do circulate body-wide. And what you see here is live in vivo imaging of these modified capsids, work done with Dr. Kathy Ferrara from Stanford. We worked with our chemists to modify these capsids so we can track them in vivo. And what you can notice is that with AAV9 modifications, you can now take AAV9 that doesn't transduce the brain well and transduce the brain, but you still have very strong expression in non-target organs such as the liver. The liver can be a big problem because it's a source of major toxicities, the reason why many clinical programs are halted and why we have those warnings on medications. So we wanted to understand more at the molecular level what's happening. PET is great for in vivo coarse imaging, but it doesn't give you the molecular identity. And here's where we recruited some other tools that the group has been working on, including whole body tissue clearing. Briefly in this work, we did use the vasculature to perfuse modified clarity reagents in an effort to clear the brain, but this resulted in clearing the entire body. This turned out to be very useful for biodistribution of systemically delivered molecules, viruses and others. And this allowed us to then look at every single organ in the body and how these modified engineering vectors might distribute. And what we noticed was that, yes, we can have very good transduction of brain neurons, but we still have liver expression and heart. And that was a problem. So we went back to the directed evolution platform and we asked more of it. We asked that not only we get vectors that are strong in the brain, but vectors that target the liver, for example. And in order to do that, we had to, again, think very hard about what kind of high throughput assays will give us this knowledge and combine with data science. So we modify the surface of the AAV capsids, mainly the variable regions, and we did this process in parallel in multiple Cre lines because this allowed us to apply both positive and negative selection pressure in a more principled way. And it gave us a list of vectors that could have intended properties. So to summarize about a decade of work, AV9, the parent for directed evolution, moved from a capsid that doesn't transduce the adult brain as strongly but transduces the liver, to PHPEB, the intermediate generation that transduces strongly the brain and the liver, to the most recent one, CAB10, that transduces the brain but not the liver. And many of the other peripheral tissues we looked at are also not transduced. This is very involved iterative engineering, at times tedious work, but it was important in order to take us to the capsids that can be most useful. And another, I think, very important and useful thing that came out of it was that in these libraries, we found, because the scale of the data and because we were able to prune with that data with our CS colleagues, we found not only vectors that can cross the blood-brain barrier, but vectors that have different preferences for cell types. So we learned that very few amino acid modifications can actually bias an AAV from a neuron to an endothelial cell to an astrocyte. And this sequence of images, I think it's quite important because it shows that with a ubiquitous promoter such as CAG, you can target neurons or astrocytes or vasculature just by the virtue of capsid modifications. Now, this can be quite important because there are many benefits of tropic capsids, and I will mention just a few. The AAV is mighty but small. It cannot package a lot of genetic material. So then if you can move the tropism on the capsid, you free up room from the bulky gene regulatory elements, and it allows you to use more interesting diversified cargo. It also, if you don't have a sink in off-target organs or off-target cell types, you can lower the AAV dose to get the same effect with less material and maybe also less immune side effects. Imagine your target being the neurons, then it's better to stay clear of antigen presenting cells in the brain. So over the years and with the NIH Brain Initiative support, we and collaborators built a toolkit of systemic AAVs for diverse systems, going from rodents to non-human primates and in vitro human systems. And importantly, the NIH Brain Initiative Armamentarium enabled not only to create these tools, but to validate and deploy them and make them available. And this is a list of where the tools that I've covered and I will cover still can be found. Through AGENE, through Cal Poly Pomona, NIH Vector Core, the Caltech Clover Center, and UC Irvine. And when looking on AGENE, you can find this decision tree that shows the species and then what kind of capsid and gene regulatory elements can be used to target different cell types. And this is also covered well in a blog from AGENE. It was very encouraging to see that after help to distribute these tools widely, they were used within neurosciences and beyond as well. And I'll show you a few examples of how these tools have been used by independent researchers. So these are non-Caltech researchers that have taken the tools and used them for their own neuroscience questions and their own uh, projects. And there's many examples, but maybe my favorite application is the now gainability for us as a community to do brain-wide genetic screens. And these brain-wide genetic screens can not only give us the pathways, the molecular pathways that underlie behavior, but they can also tell us how to maybe course correct in the disease case. So there's beautiful publications coming that highlight this ability to better understand the pathways in disease and to correct them. Within my own group at Caltech, we've also applied them, and I'll give you just one unpublished example today. This one, I think, is important because it shows the value of combining targets. The brain lives within the body. They talk to each other, they influence each other, and many disorders of the nervous system, they're central and peripheral. And this is the case of Friedreich's ataxia, where there is a loss of frataxin, and the loss of frataxin is in many tissues, but it's important to correct the central manifestations and cardiac symptoms as well. It pairs well with AAV gene delivery because the frataxin gene, the code can be packaged. And this is a project that was driven by MD PhD student Acacia Mayfield that just graduated from Caltech last week. And when Acacia was pursuing this project, she realized how important it is to have the best vector for the brain and the best vector for the peripheral nervous system. So that's where we needed to take a bit of a detour and apply the same methods that I mentioned that we applied for the brain to apply them instead to the peripheral nervous system. And you can see here now vectors, again, evolved from AAV9, that with modifications, you can evolve them to reach no-dose and DRG much better. No-dose for the vagus, DRG relevant for pain, with distinct liver profiles. So then Acacia was able, collaborating with her colleagues in the lab, was able to go back to the project and take the best capsid for the Friedreich's ataxia project, and chose CAB10 for brain targeting because strong brain, weak periphery, and then chose PNS2 because of the strong expression in peripheral tissues relevant to Friedreich's ataxia. And then she applied these tools to an inducible model of FA and run the cohorts through a standard behavioral panel. And this was a lot of work. I'll show you just a few examples of how with co-delivery of frataxin, Acacia was able to rescue behavioral phenotypes in mice. So you see, this is a narrow beam walking assay, and you see the wild type crossing without any problem. Then you have the FA model in the middle that has slips and falls. The fall is very minor, it's just the cage. That's and then with the rescue, you can see much improved motor behavior. Of course, it's hard to get back to full health, and you see that difference in mouse weight as well, but the locomotion is much improved. And of course, Acacia did this in multiple animals. And this introduces us to this concept of combined gene therapy for multiple targets. And once we move beyond monogenic disorders and beyond one organ disorder, we do need to embrace these modalities. And it's important to mention that Acacia did run the independent cohorts, central rescue only, peripheral rescue only, and learn that both are needed for rescue. Because Acacia is training to be a physician that hopes very much to be able to prescribe the therapy she worked on at the research stage to her pediatric patients, maybe in a decade or so, she always reminds us in the lab that we cured enough mice by now. So what about primates? And we can see that there are big evolutionary gaps from the rodent to maybe where we need to be with human disease. So the lab did work over the years on trying to address this problem. And this is one example. So here there's no blood-brain barrier involved. This is a slice. This is work done in collaboration with the Allen Brain Institute. We are fortunate to have access to their in vitro preps of macaque tissue, macaque brain tissue, and human brain tissue. And then we were able to put the AAVs and see, are they transducing better? So you can see that even without the blood-brain barrier, the ability of transduction for modified vectors is much higher. And indeed, these modified vectors can also cross the blood-brain barrier in the marmoset, a small new world non-human primate. It's more distant than its relative, the macaque, which is here. So in the macaque, we worked with UC Davis and our longtime collaborator, Dr. Drew Fox, and Drew found our posters at one of the Society of Neuroscience conferences and then the Brain Initiative meetings, and convinced us to start working together in making vectors that can cross the macaque blood-brain barrier. And this is one example of a rainbow-like macaque brain that it's not transgenic, it's through delivery of vectors through the bloodstream. And this allows for the ability to also achieve genetic type Golgi-like labeling. In pictures, it's hard to show the extent of brain coverage. So if we go through cleared volumes of marmoset or macaque brain, you can see the extent of coverage. All of this is non-invasive, and Drew told me that to do this for a macaque through direct injection, you need an operating room, lots of money, you need a neurosurgeon. This could be done through a simple IV injection, and the brain had all the transgenes necessary. Interestingly, through this work, we learned something very important back. This was tool generation work, but we learned that the blood-brain barrier varies across strains and even within species. And the first experimental result that showed us that was a test of the PHPB across mouse strains. And this result, as a basic scientist, is very exciting, like it's what's going on here. With a translationally minded read, this is very concerning, because what you see here is the modified vector performing very well in C57, intermediate in other strains, and not at all in BALB/C. And I cannot emphasize how important this is, because these are highly inbred laboratory mouse strains, as controlled as possible, and we have these very big differences. And if we think about us in this room, by virtue of our many biological variables, it is likely that our blood-brain barriers are going to be distinct. And the reason this is important for gene therapies using AAVs is that once you get an AAV dose, the body can create neutralizing antibodies. And if that vector is not matched to your BBB biology, it might not work. And then because of neutralizing antibodies would be harder, would make it harder to get the therapy that matches later on. So this was, needless to say, something that we really wanted to understand. And in the case of PHPB, the receptor insight came from multiple groups as the Ly6A. And that explained all of the behavior. Ly6A is defective in its membrane anchoring in BALB/C. So we knew that if we know the receptor, we can say whether this would work across different species or across or strains. So that's why we've started to look at all of the possible variables that within reach, such as age, disease state, sex, and ancestry. and try to understand more about how the blood-brain barrier might vary across these brains, and importantly, even within brains, because the brain itself is vast and not all vasculature is the same. There might be different vascular signatures in frontal areas versus cerebellum, for example. However, the bottleneck is how do you figure out all of these receptors in different brain areas, in different people, and of different biological variables. And we realized 6-7 years into that we do not have to guess anymore or go at this approach one by one, because the community, so beyond our lab now, there is thousands and thousands of interesting AAVs with interesting properties from other academic labs and from industry as well. Industry is generating very, very strong solutions. So then what we have to do is to take the strongest, most interesting vectors and just ask them how they get across to figure out the molecular pathways. And by asking them, of course, the AAVs won't talk back, so we had to design in vitro systems where you can expose candidate surface receptors, apply the AAVs, and look for matchmaking. And this now can transform the studies of BBB penetration into receptor matching systematic experiments. And this approach has started already to give us new receptors beyond transfer, beyond the very small set that we knew as a community. In the past 2 years, we're populating this list with many receptors, and some of them, importantly, they're rather surprising, because you wouldn't go to an enzyme to look for transport of the AAV, but one of the engineered AAVs found as its best match the carbonic anhydrase 4. It's an enzyme. It's very important in blood pH regulation. Is next to the bicarbonate transporter, and importantly, it's highly conserved. It's part of the Rubisco pathway in archaea. And we learned that it can mediate transport across the blood-brain barrier. We included it, and this was work driven by it's a bit cut out by Dr. Tim Shea, that also has a poster at the meeting, and he's the scientific director of the Clover Center at Caltech. So Tim included CAR4 in the screen because it came as enriched in our single-cell RNA endothelial surface molecules. And it's interesting to look at the pattern. So in a wild type, you see how abundant CAR4 is on the vasculature. We were fortunate that JAX already had knockout mice for carbonic anhydrase 4. So then we put them to the test. And when you apply a CAR4 binding AAV to the wild type, you can see very strong transduction. If you remove CAR4, there's no transduction, while a control virus that binds Ly6A can transduce both the wild type and the knockout. One reason why having more receptors is important is that they will have different preferences in different organs and different cell types. And I want to highlight when looked at the transcript level. So you have CAR4, transferrin, the more recent one, CD98, it's very promising. Here's the publication. You can look and see the RNA transcript level. And importantly, as we discussed, you can see the liver profile. Carbonic anhydrase is much lower in the liver while still having strong expression in the brain. So this is just to say that more receptors is better. And that's why Tim worked on with Retrogenics to design a high throughput screen of thousands of human membrane-associated proteins. And this pipeline now is generating more and more interesting receptors. We found the hits, then we use biochemistry, SPR, optical measurements of binding of AAV and receptor, and as we funnel through the strongest candidates, then we go through in vitro internalization, validation, and then in vivo. And this path has already generated other receptors such as LRP6 in a preprint here. So importantly, this just by going through all of the interesting AAVs, we can find new blood-brain barrier receptors, but also beyond the blood-brain barrier, you can find receptors for the peripheral nervous system. There are very difficult targets, and surgically they're sometimes even more difficult than the brain, because there are AAVs that target different organs: the lung, heart, pancreas, kidney. And here's an example of vectors that target the enteric nervous system, for example, in non-human primates. Now, what do we do with this receptor knowledge? We're populating the receptor list, but we've started on the receptors that we know already to understand how they interface with the AAVs. So through structural work and AlphaFold docking studies, we've been learning a lot about the interface of engineered AAVs and their receptors. And this now allows us to bypass the limitations of blind screens, the way we've been doing now, and engineer viral vectors against the receptor target. That's one. And I don't have time to show you that work. But you can take the libraries and you can do all of this work in vitro. So it's important for non-human primates, it's very important, because in vivo NHP screens are very slow, very costly, and that's why it's hard to make progress. However, if you know the receptor, you can look across conserved sites and modify AAVs to bind the conserved site across species. And this allows you to go into the NHP experiment with some level of confidence that this will work, because the mechanism, at least the mechanism, checked. And similarly, down the road, this could also allow to do this for human clinical trials. So that's why receptor-targeted engineering is very important, and it's something that's it's an emerging and fastly developing field. However, with viral vector delivery, there are some caveats. I already mentioned that it's a small virus. You cannot package a very large genetic material. It's sometimes hard to even package gene editing machineries if you want to use the largest editing enzymes. And in addition to that, much of the population, especially later life stages, has neutralizing antibodies just by virtue of living long enough. We're exposed to AAVs. It's a non-pathogenic virus, so we might not be aware of it, but we can have blood work done and see does one human have neutralizing antibodies or not. This is a strong reason for exclusion from clinical trials if one has high level of neutralizing antibodies. That's why it's important to move beyond viral vector delivery, in addition to some of the immune challenges that viral vector delivery could pose. So we've, in preliminary work, and I'm excited to show this to you just as a starting point, by knowing new and diversified blood-brain receptors, we can look at the structural information of those receptors and understand how can we engage them with small molecules, with peptides. How can we engage them to now load these receptors with different modalities, whether they're protein-based modalities, antibodies, or oligonucleotides. And we weren't sure this was going to work, because when you engineer an AAV, you modify the AAV surface up to 60 times because of the repeat of the viral protein. So it might be the case that in an AAV you have some avidity effects and amplification effects. So it's not as straightforward as getting the peptide from the AAV and now trying to load it with, let's say, a PET label. However, at least in a few cases, we already showed that you can take, for example, CAR4 that we showed can enable BBB transport and load an antibody. And you look here, the unmodified antibody, so the one that doesn't bind to CAR4, 2 days after systemic delivery, you don't see much expression in the brain, while the modified antibody has very strong presence in the brain. Interestingly, for this experiment, we chose to use atezolizumab that's used in the clinic for non-small cell lung cancer. And it's important because many of these patients do have brain metastasis, and the antibody works in the periphery for tumor clearance, but cannot easily cross the blood-brain barrier to tackle any brain metastasis that might happen. So because we use this antibody, it can already enter neurons. So if we look at high magnification, you can see the non-modified product. You don't see strong expression in vasculature or neurons. So in green, you have the new end stain. So green is a marker, it's a neuronal label. And in red, you have the antibody. And you can see that with the modification, you see the product in the neurons. So this creates the possibility of not only enabling blood-brain barrier crossing, but once you cross, you could target it to specific cell types. But we can only achieve this dream if we have enough receptors with the needed properties, because you need both receptors that are present only on the blood-brain barrier or receptors that are present everywhere, so the way transferrin is. And you want them to have varied biodistribution for different organs of interest, and in a way that they can engage different cargo, whether it's RNA or medium molecules or protein-based. And just the two receptors I mentioned today, the CAR4 and LRP6, they already show the power of diverse receptors, because CAR4 is mostly present in vascular cells, which makes it the candidate if you want specificity, because you can engineer a bispecific antibody. One arm crosses the blood-brain barrier, the other one enters the desired cell types. While on the flip side, there's LRP6 that's present everywhere. So if you want the ubiquitous delivery, you would go for the LRP6 engagement. In addition to delivering research tools and therapies, the importance of identifying blood-brain barrier receptors is that we can better anticipate and block pathogens. Imagine now, and it's good it didn't happen, and I hope it never happens, that the continuously evolving SARS-CoV-2 evolves to lock into one of these receptors and get to the brain. It would be good to know the receptor and be ready with blockers. And this is not just a theoretical possibility, because there are pathogens that cross the blood-brain barrier, including HIV. This is a publication that shows how bacteria could evolve to use the transferrin receptor to access the brain. And now our ability to anticipate threat and be prepared for it is enhanced because of the ability to go through genome evolution. So this is just a simulation of how SARS-CoV-2 would evolve and how the list gets populated with different strains. So if you have the ability to do this, and then you have a very long list of all of the blood-brain barrier portals, you can look for matches. And if you find theoretical matches, then you can be a bit ahead of the game and use all of the developments, especially in structural biology now, where you can be prepared with blockers. And we've done this mainly for engagement of these receptors, but you can flip it and have blockers, where you can make in silico a list of peptides that can engage these receptors. And the algorithms will tell you these are worthwhile testing in the lab. And then we took a few, we tested them in the lab, and that's how we were able to engage some of those receptors to carry antibodies and the like. And this can expand to mini proteins as well. So we can take, importantly, as I mentioned, the 7 mers from AAVs don't always translate to other modalities. But it turns out we can take the 7 mers from AAVs and scaffold them and produce them separately. And that's fast forwarding the way we can engage these receptors, because we can make them and see which ones can actually engage the receptor. So this brings me to to closing that understanding the blood-brain barrier transport biology can help in a few ways to deliver brain therapeutics of diverse modalities from the blood, non-invasively from the bloodstream to the brain. And it can be oligos, antibodies, nanoparticles. And with clever engineering, one can also aim for cell type specificity or brain region specificity. At the same time, it might prepare us to better navigate health threats from bacteria, viruses, and tumors. And it's important that this is done in a way that's both understanding and respectful of biological variability. There is biological variability, and it's important. It's important to accept it, to know it, and to work. So this knowledge and down the road, maybe preclinical and clinical insights can help all those in need. And the dream goal would be that now we can leverage these blood-brain portals to understand the brain better and also to target it with therapies. And this can be done in a way that can access the needed regions and brain cell types throughout the brain. The flip side I mentioned briefly, by knowing the receptors, we can anticipate threats and be prepared with blockers against viruses or bacteria. And we can do this across biological variables as imposed by evolution and across lifespan as well. The paradigm that we've piloted for the brain can also be now pointed in a similar way towards other physiological defense structures, such as the placenta, to access the developing embryonic brain. Many neurodevelopmental disorders post-delivery, they're quite advanced, so it might be important to start thinking about that significant barrier. And also for skin, which is our biggest protective barrier. We should be very grateful and take good care of it. So I want to thank you, and I have a few things. And of course, the team, you've seen their names, you've seen their work. This work is slow, difficult, a lot of sweat and blood. And they are so brave and don't shy away, and they just get it done. And I'm so grateful to my group at Caltech that I started the group a little over a decade, and the combination of engineers, data scientists, and neuroscientists, they really learn from each other and they help each other. So I'm very grateful to them. And I'm very grateful to the NIH Brain Initiative for getting us to this point, for allowing us to pursue some dreams. I didn't start my lab as an AV engineer. I started as an opsin engineer and neurotechnologist working on Parkinson's. But because of the Brain Initiative, I was able to take the learnings from opsin engineering, pair them with directed evolution with my chemistry colleagues, and work towards the AAVs that you've seen today. And in terms of the translational part and clinical, there is one gentleman that I would like to thank, the NINDS director. I don't know where are you, Walter? So the hiding in of course it's on type. He's very humble. Dr. Walter Koroshetz, the NINDS director, approached me when I was just starting my lab. I didn't know I can do much for neurology, and invited me to this very clinical meeting in San Diego where the room was full of doctors. And I was sitting there thinking, what am I doing here? And then after I gave the talk, the room told me what I need to do, and then I did it. So thank you, Walter, for your almost 2 decades of service for the NINDS. And of course, the Brain Initiative for all of the support and guidance along the way. So I will pause with this summary, and thank you so much for your time and attention.
Thank you, Viviana, for sharing this amazing journey, which is nowhere near over yet. We have time for questions, both from folks in the room as well as from folks online. So please feel free, if you're here in the room, come up to the microphone. Those of you folks online, type them into the chat and we will make sure that they are read out.
Is this on? Yeah. Can you say a little something more about the off-target toxicity effects of these viruses? That to zero order, I would have thought, who cares if the liver gets a little bit of green?
Yes, that is a very good question. So it turns out that everything that's delivered systemically gets filtered through the liver. And if there's too much AAV, the immune reactions can be to the capsid and the cargo. And we know for a fact from mice experiments, but also unfortunately clinical work, that liver toxicity is a cause for disease and even death. So what happens if you try to inject a rodent at a very high dose? We cannot go to a very high dose without having a very high level of liver enzymes. So you can measure liver enzymes in the bloodstream, and that tells you what's the safe dose. And what we learned that with the modifications to the target for the liver, we can safely increase the dose, or we can get the same brain expression because you don't have the liver as a sink. So the liver is a problem for two reasons. If the brain is the target, why would you waste so much hard to make and precious AAV on the liver? That's one manufacturing access, maybe cost reason. And the other one is the serious toxicity problem. However, we have experimental evidence that we can detarget it, and this is better overall for the amount of product used and the health of the animal, because we don't detect these inflammation markers in the bloodstream.
Viviana.
Yes, I hear you well.
Okay. I'm standing, yeah. I wonder if you could tell us a little bit more about the host variables in transduction efficacy. So you mentioned that in children, this AAV9 virus worked well, but not in adults. And you also mentioned some specific targets, carbonic anhydrase. Are these, for example, regulated by sex? Are they heterogeneous across different populations? Or are there multiple viral targets that show this heterogeneity? What should we expect in terms of the general applicability? Do we have the right targets to be able to predict that, or do we just need to check a lot of things?
I think we have classes of targets, and that's very helpful because it's not as difficult as one human, one receptor, one AAV. We are, and here's where I step back, not necessarily back from my statement, but we are more alike than different, okay? But there are some differences that matter when it comes to disease. And the behavior of these receptors correlates very strongly with the transduction. And we know a few things by doing single-cell RNA across lifespan. There's data for Alzheimer from Andrew Young, now at UCSF, working with Tony Wiscoray. Before that, there's data sets where you can look at the receptors on the BBB staying flat across lifespan in disease or going down. We looked at the CAR4, and we were very happy to see that the expression is constant in this data set. So that's one example. The other one, we know that by sex, male and female mice deal with liver processing differently. So when we inject in male and female mice, we do see different level of liver transductions. We didn't have necessarily the support and the bandwidth to pursue this side by side, because it would require not only looking at the sex variables, but the estrous cycle. So you need good markers and to monitor that around the estrous cycle. So we know the big classes that matter. Age is also a variable, because the younger the mammal, it might be the blood-brain barrier is more permeant. versus later, but also the counter argument can be made the more mature the mammal, the blood-brain barrier might also be leaky due to disease, as the very big one is in stroke, right? There's no barrier. And there are some areas in the brain that do not have a blood-brain barrier as well, subfornical organs. It's a very long list and conversation, and I hope we will start to prioritize and to understand the blood-brain barrier better, because it matters in terms of delivery. And we have all of these wonderful research tools from the NIH brain community, and many of them we cannot deploy in non-transgenic animals.
Hello, my name is Dr. Cheryl Lohman. I'm a local physician here, was before I retired due to disability. I'm also an electrical engineer. This whole conference is absolutely fascinating to me and what everybody is doing. My comment is this. On your first slide that showed the different disease processes that involve the brain, one big disease process I saw missing from that slide is long COVID. This is new information. 20 million patients, which is more than patients with cancer, having a $3.7 trillion effect on the economy. And there's no funding in the NIH right now, but Bernie Sanders is working on that, on his moonshot. And I'd just like to let people in this audience know that so much of this research that is so amazing and so groundbreaking could very easily be applied to the problems of neurological symptoms of long COVID. And if you wanted one particular example, it would be neuroinflammation. Thank you.
Thank you so much. And that's how my slides keep evolving with insight and questions from the audience. I will add that. And what we know about long COVID is there's no, to my knowledge, clear evidence that the virus crosses. However, the aftermath of the virus can. Particles, inflammation, peripheral inflammation will make its way through the brain. And I like very much what you said about how this neurotechnologies from the Brain Initiative developed for brain disorders actually can be applied in a broader context for cancer, for long COVID. And one example, and this is for tissue clearing, not for the AAVs or optogenetics, but when I gave this talk to my colleagues in biology division at Caltech, it's a very broad division going from microbiologists to human brain-machine interface researchers and engineers like yourself. One of my colleagues, Diane Newman, she's a geologist and microbiologist, saw the tissue clearing, said, oh, this is great, we can apply it to a problem in cystic fibrosis. And at first I thought, oh, maybe I didn't explain well because it's all dead tissue. But what her group ended up using tissue clearing for was to quantify the progression of bacterial infection in cystic fibrosis and the response to the antibody by looking at ribosomal RNA in sputum from cystic fibrosis patients. And the learning from that was that the estimates were from prior single cell sorting were not accurate, and that was misguiding the antibiotic regimen. So tissue clearing through clarity that came through the Brain Initiative and applied to cystic fibrosis. So for sure there's room to expand.
Yes, Dr. Gradner, we have a question from the online audience, and it's asking if you can comment a bit more about what we do and don't know about BBB biology. So for example, how dynamic are the receptors that are found there? Do different environmental stimuli change the composition of receptors? How heterogeneous or homogeneous are they in terms of kind of their spatial distribution?
Yes, it's a very good question, and unfortunately, we do not know enough. We do know that there are changes, but we do not have the data sets to look across lifespan, across disease states, across ancestry. And that is very important information to have in order for us to match the delivery vehicles with the blood-brain barrier biology of the patients in need. All of that, I think, will emerge because there is an accelerating effort from many groups, and I should mention a pioneer in this area, Dr. Shenghua Gu. She was an NIH Pioneer Awardee for studying the blood-brain barrier, so very important work. So there is work, it's just much more effort is needed.
Hi, Viviana. Great talk, as always. I was wondering if you could elaborate on what you find the major translational hurdles to the clinic for these, any one specific vector or any of the concepts you brought up today.
The main hurdle is that we cannot test in humans. The result will be known once you go into a phase 1 safety clinical trial in a small, well-staggered, thoughtful way. And this is, for me, it's very, it's a big item that I don't have many things that keep me up at night because I need to sleep to do the work. But one thing that keeps me up at night is like, what if we cause harm? And this actually motivated the work on the mechanism, because the only way to decrease that risk to a level that's worth it is to know the mechanism. And once you know the mechanism, is the receptor there? If the receptor is not there, don't touch it, because it's not going to work. If the receptor is there, is it anchored to the membrane? Right? Is the site we're engaging a conserved one? Because for carbonic 4, interestingly, some AAVs engage non-conserved sites. That's why they work in mice and they don't work in monkey. However, on the CAR4, there are sites that are conserved, so you can engineer against the conserved site. Because CAR4 has 70-something% homology. LRP6, more than 90% homology. So then it's much easier to find these conserved sites. So it goes back really to the basics. Proteins, structural biology, protein-protein binding, chemistry, medicinal chemistry, like really to the just drawing board. And we know what questions to ask to minimize the risk now. It's a matter of doing that work and refinement. So when you go into a phase 1 clinical trial, you've checked everything that is checkable.
The boss has the last question.
Hey, Viviana.
Okay.
Thanks so much.
The hard one.
Well, I was just wondering, is it futile to try to use your technologies to create AAVs that escape the immune system? Because that's one of the bigger problems, as you mentioned. Also, for people who are at the pioneer stage and kids who are getting the treatment for the first time, and maybe getting a dose that's not effective, and you would like to be able to give another dose, or some of the gene therapies coming out now with AAV, and they're not that great, but there might be a better one in the future. Or is there a way in which you can redose, whether you can use your technologies to allow that to happen?
Thank you, Walter. This is maybe the biggest problem in AAV redosing, because at some point it might be needed for the reasons you mentioned. And I have two things to say. One, that we've tried so many approaches and failed enthusiastically many times, and we keep trying. That's like the summary of it. However, there's some hope. What we learned was that in some cases you can move the peptides from one serotype to another serotype. So for example, we can move it from AAV9 to AAV1. The levels of neutralizing antibodies are different against different serotypes. So if, let's say, a patient has AAV9 neutralizing antibodies but not one, you could do this serotype switching for redosing. And that we know it works in mice, and that can be one powerful approach. I think it hasn't been explored quite as much, but that's one path. The other path that we've taken and now I'm less optimistic about it, is to use directed evolution to modify the binding sites for the neutralizing antibodies, right? Because that would be the theoretical path. And we were doing that for a while until there was a publication from the SMA study. So I think preprint or from the SMA study, I think from a Novartis team, the blood of patients that have gotten the SMA therapy was studied for neutralizing antibodies. And what was found was that it's a very rich, complex immune response that's against many sites on the capsid, against so many sites on the capsid that if you are to try to modify all of them, you just crush the virus. So that kind of made me less optimistic about this approach. We're still trying, but it's hard to engineer 60 sites. Now we're doing 120, let alone because they repeat on the capsid. So to modify the capsid enough to evade the complex immune and polyvalent immune response while still making a gene delivery vector, it's a bioengineering problem. So we'll see where it goes. But serotype switching works well, and maybe there's other methods of shielding temporarily or immunosuppression short term upon start of the trial.
Terrific. Please join me in thanking Dr. Grodno for a spectacular talk.